And this slide here puts in writing or in sliding or whatever you want to call this particular presentation, the point I've made that clustering, the way it's often presented, is actually trying to find clusters, namely groups of points which are clearly separated, and then you want to actually identify those clusters. However, another important use of clustering is in classifying. And when you use clustering for classifying, you're just trying to find a compact set of subregions so that when you find that the point is in a subregion, you know that the other points in that subregion are near it. And that's a classification problem. And it doesn't matter, that particular use of clustering doesn't really matter whether you get different clusters when you do it different ways, because we're not trying to identify the unique clusters. We're just trying to find geometrically compact subregions, and that can clearly be done in many ways, as in fact we showed in the 1,000 points, which had different ways of dividing into 3 clusters, which were quite different globally, but they all satisfied the criteria of having 3 compact subregions. There are other technologies here called latent factors and mixture models, which have the same type of classification goal. They're related to the clustering algorithms. They're mathematically pretty similar. And in fact, both of them can use annealing if you wanted to. And the latent factor methods are, for instance, used by Google News to group, at least I think they claim to be used by Google News, but in some place I read, to group articles together which were on a common topic. Another point I wanted to make about clustering, which we'll actually illustrate very soon, is that we have for clustering 2 major types, or maybe more than 2, but at least 2 important major types, are those that work in real vector spaces, where your points are actual vectors, and those where the points are not vectors. Now, you remember in the case of item-based collaborative filtering or user-based collaborative filtering, I pointed out that those spaces are not really vector spaces because you only know some of the components. Those are examples of spaces where you can define a distance, but you can't really define vectors that you can add. Because when you have a vector space, you can add vectors together. In fact, that's how I found the center of the cluster, was to add the vectors together and divide by the number of points. That does not work unless you have real vectors with explicit components in all dimensions. So there's a very important set of problems, which are actually probably the dominant problems used in e-commerce and lifestyle informatics, where the spaces involved do not have proper vectors, but they do have metrics. So now we do some examples just to show you the different types of clusterings you can see. The first is from mass spectrometry, and then where there are actual peaks, because those are 2-dimensional vectors, and so the mass spectrometer peaks, and they have real clusters, and they're clearly separated. And there's genomics where they're also separated, but they're not in 2 dimensions. And so that's a case where you need to use, actually you can't use k-means because k-means only works if there's a vector space. And I have a PlotViz for the genomics example. Here's the example of the peaks. Each of these little points here is a mass spectrometry peak. And they're actually so-called sponge peaks. Those are the case of isolated peaks where there's just one point. Those are these little triangles. And here we have actual clusters, and the clusters have centers, which are these triangles. And if you look at this case here, you can see that this data is, these clusters are actually separated. They're a bit ambiguous, like these points here, whether they belong to that cluster or that cluster is not so easy because they're sort of unclear. Probably they belong to this cluster, but the algorithm assigned them to this cluster. That's often true in clustering. They're bound to be ambiguous points, which at the edges of 2 clusters, and which cluster you assign them to is not well defined. In the classification, I mean, it will not make huge differences if you analyze the data carefully later on, but it has to be borne in mind. So this is the original clustering application. We have a whole bunch of clusters which are clearly separated, and with a little bit of ambiguity here or in here where clusters are near each other. So that's two-dimensional vector, and this is using this deterministic annealing-based enhancement of k-means. And this particular data sample had around 30,000 clusters in all. So this is a tiny sample of the space. Now we come to some metagenomics examples. These are genomic sequences where you can define the distance between the sequences, but they're not vectors because they're just streams of characters, and the length of each stream is different. And there's not easy to do so-called multiple sequence alignment, where you actually align all the sequences to make them actual vectors. So you tend to analyze them without doing that. But you do see here some rather pretty, in these metagenomics cases, clearly identified clusters. You can ask whether this one here is 2 clusters and things like that. And there is some distortion. These clusters are not circular. They tend to be elongated. And we could discuss why that is, but that's for a different time. They're not needed here. This is called deterministic annealing. That's the type of clustering. BW is pairwise, meaning it's not vector. You just have pairwise distance, and C is clustering. Here's another set of genomic sequences for a particular sample. And it's a bit much messier, but you can see enormous amount of structure with clear separation between them. Sometimes there's some lack of clarity, like there are lots which are joined together or near each other, but there's lots of separated clusters here. Another example here, we actually have 440,000 sequences here, which is actually a mix of clustering and interpolation. We can do it more accurately now than when we did this. And this is not fully clustered because there's over 100 clusters, 120 clusters or so in this data set. And this particular clustering algorithm allows us to stop as we start with one cluster being the whole data set, and we gradually chop it up in a hierarchical fashion of parts. Here we stop when there were only 7 parts. And here's yet another example where, again, we have, here you can see whether this is a cluster or it's just a sort of scatter of points over here. Here we have really distorted clusters, long, thin things. Technically, the length of these, the elongation of these clusters is due to the fact that the sequences in them have different lengths. If you take sequences of the same length, they tend to be much closer to each other than if you try to look at the distances and incorporate sequences of a variety of different lengths.